Good evening, everyone, and a very good evening to you all, and welcome to our first London Lecture of 2020. It's heartening to see so many alumni in the audience tonight, along with new faces as well. And a big welcome to those of you tuning in via the Facebook live stream. My name's Dylan Edwards, and I'm the Pro-Vice-Chancellor for the Faculty of Medicine and Health Sciences at UEA. Our speaker tonight is David Lea-Smith, a lecturer in microbiology from UEA's School of Biological Sciences. You may be wondering why then it's me as Pro-Vice-Chancellor for the Faculty of Medicine and Health Sciences rather than Mark Searcy, who is the Pro-Vice-Chancellor for the Faculty of Science. Well, Mark couldn't be here tonight, but also I have a particular connection to bio, I was a member of bio for 18 years, from 98 till 2016, something like that. And I was head of school for 5 years there before switching over to medicine and health sciences. So I've got a big connection there anyway. I, in fact, left the school at the time that David joined and I took up my role then. But I've been chair of cancer studies in the school and then subsequently in the medical school ever since. So before we hear about David's research, I wanted to give you an update on some of the things that have been happening recently at UEA, in the sciences in particular. So in October last year, our brand new science building was formally opened by world-renowned scientist and activist, Dr. Jane Goodall. And here you see her at the opening ceremony. Dr. Goodall led the Vice-Chancellor and invited guests in a chimpanzee greeting before touring the building and seeing staff and students at work. The building is used by staff and students for the study of environmental sciences, computing sciences, physics, engineering, chemistry, pharmacy, and of course, biological sciences. It is the home for the teaching labs for the whole of the science faculty. I wanted to point out some of the architecture. I do encourage you to come up to UEA and have a look around and see the pace of change that has happened there. But the architects in building this building, which incidentally everybody likes who goes in it and uses it, I would say, they've tried to echo some of the old Lasden 60s buildings in there with the horizontal panels of the floors in the building. The steps down to the entrance area are meant deliberately to evoke the steps in the square and the community sense that builds there. And the pillars are reflective of the pillars in the library area. So it's been carefully thought about to actually evoke the spirit of UEA in putting it together, this new building. This is a big social area that's just inside here. There's another, just to where I'm over here, there's the Julian Study Centre. So this is a big student social milling area around there. So it's a really great building. And not only is it packed with state-of-the-art scientific facilities, it's also one of the most accessible buildings, university buildings in the country. UEA's Access All Areas network, the university's forum for staff and students affected by a disability, were consulted throughout the project to ensure maximum accessibility. So we've also just celebrated 1 year since the appointment of Ben Garrett as Professor of Evolutionary Biology and Science Engagement to the School of Biological Sciences. Professor Garrett appeared on BBC Two last month in Baby Chimp Rescue. I hope you all tuned into that. It was a three-part series following the life of orphaned baby chimpanzees, particularly in Liberia, as they adapt to life in a rescue compound. It's a fantastic series, really very, very emotive. And Ben's been on campus over the past few weeks teaching science engagement to undergraduate students, taking the science communication module, and here he is actually doing some stuff in the lab, in the teaching labs there. In recent years, our climate change research has contributed once again to the global carbon budget. The global carbon budget was instigated by UEA's Professor Corinne Lequery as part of the Global Carbon Project, a community effort to provide timely delivery of carbon research and help stabilize the concentration of CO2 in the atmosphere, which is the main driver of climate change. An international research team featuring UEA scientists has shown that last year's emissions are likely to be 4% higher than those in 2015, the year of the UN Paris Agreement, with a decline in coal burning that's been offset by strong growth in natural gas and oil use worldwide. Responding to the findings, Professor Lecarrier has called for stronger policies that are targeted at phasing out the use of fossil fuels, and we'll see if any progress has been made on this by November, when we will welcome the UN Climate Change Conference, COP26, to Glasgow. I should say as well that this is a very active thing, given UEA's history with climate research and being in the forefront of identifying the impact of carbon on climate change. We take this particularly seriously, and tomorrow, in fact, we, as part of a series of what we're calling Big Shift Big Days, we have a Big Shift Big Day on sustainability, which should be addressing really how the university responds as an organisation and what we do on campus to live up to the goals of achieving carbon neutrality. We've signed up last year as well to to recognition of a climate and a biodiversity emergency. So we're articulating how we address those emergencies. And we've also become a member of the EAUC, that's the Environmental Association of Universities and Colleges, which is working as bringing all of the universities of the UK together to address in a one-year mission to deliver real impact on energy and carbon usage and sustainability, and we'll report at COP26 in Glasgow. So looking to the future, we'll all be lending our collective voices to the course for action at COP26, backed up by research from across all of our faculties. We'll continue to respond to the government's grand challenges in partnership with local, national and international organisations who enable us to take our work further. And we'll continue to support research with the potential to change the world we live in for the better. Like that of Dr. Lea-Smith and his colleagues from Bio, which I'm very much looking forward to hearing about this evening. David earned his PhD with Monash University in 2007, after which he moved to Edinburgh to take up a postdoctoral position investigating gene therapy strategies for cystic fibrosis. In 2009, he moved to the University of Cambridge, where his research led him to focus on understanding cyanobacteria and their role in the environment. He continues this research and leads the Lea-Smith group at UEA, whose cyanobacteria research has led to a number of important and high-impact publications and a great deal of press coverage. So please join me now in welcoming Dr. David Lea-Smith to give his talk on how algae help bacteria clean up oil spills.
So thank you, Dylan, for that kind introduction. I'd also like to thank you for actually coming to this lecture. So when I agreed to actually give this seminar, my main concern was that no one actually turned up. So it's quite pleasing to see that there is quite a reasonable sized audience here. And the title I chose up here was left deliberately cryptic in order to hope that people would actually be interested in trying to work out what this all means. So I'm going to focus on a number of different issues, starting with oil spills and then the type of bacteria that help us clean up oil spills. This is a kind of schematic of what might be happening. And then how algae actually help these bacteria clean up oil spills. So I'm going to start on perhaps a more positive note than the introductory speech, which is that I think there is a lot of signs of progress in combating climate change and carbon emissions. I've just given some examples up here. So I think it seems likely that electric cars are going to become dominant in the next couple of decades. And of course, this is going to decrease the amount of pollution from old-fashioned combustion engines. And this has been driven down by the increasing quality of these cars and the reduction in the price. Another example of this is solar panels, which keep getting cheaper every single year and no longer really rely on subsidies. And I think wind power in the UK is becoming bigger and bigger, and it's quite remarkable how this has expanded over the last decade. So this is an example of the East Anglian project between, I think, or the Greater Anglia project, I think it's called, between Great Yarmouth and Ipswich. And once completed, and they can actually set up one of these wind turbines in a day, which is just remarkable when you think about it. When completed, it will generate 3.4 gigawatts of energy, and if working at full capacity, it could meet roughly 10% of the UK's energy needs. And of course, this will be nice renewable energy. And then, of course, there's been technologies which have reduced our energy usage, and I think LEDs have been perhaps the greatest example of this. So, of course, LEDs use about 10% of the power of old-fashioned luminescent globes and have made a massive impact. So, in my opinion, the way we actually combat climate change is the development of technologies. People can protest, we can have Swedish teenagers telling us what to do, but ultimately I think governments want to actually do this, but they need the technologies to allow us to sort of reduce our pollution, but maintain our lifestyle. And so to me, the real heroes of these type of stories are individuals like these three gentlemen, and they were key for the development of white LEDs, which are really of a high standard these days. And for that, they won the 2014 Nobel Prize, which was well deserved. And this is the kind, these three individuals have probably done more for the environment than anyone else who I can think of. But, and this is a key factor, is that the world still uses a lot of oil. And I'll show you a graph on the next page. So, of course, we use oil to fuel our cars. Hopefully that will decrease in the next couple of decades. If you own a private jet, you need your fuel for that. You're not going to be able to have an electric jet. Any time you fly, of course, all the jets that the rest of us take, these require fuel. Fuel is also the basis of our entire chemical industry. So if we want to move off oil, we have to find substitutes for the way we produce chemicals. And for those of you who think this cartoon might be a little cruel, it's actually probably not cruel enough. And I think the situation's actually much worse than that. This is from last year, but I think it was the same this year. And so what I'm going to show here is there's a lot of statistics about how much oil we're using. So this is just one. And what you can sort of see is in the last couple of years, the amount of oil we produce has been increasing, as has our consumption. Now, hopefully, this will begin to go down over the next decade and gradually decrease. But basically what this means is that the world pumps a lot of oil and it transports a lot of oil. And a good example of that is there are about 10,000 oil tankers in the world, so it's about 37% of the global fleet. So there are a couple of issues of this apart from the pollution. And the fact is, if we produce so much oil and we transport so much oil, there will be accidents that happen. So there's been actually relatively few, considering how much oil we do produce and transport. This is perhaps the most famous one, I remember this as a boy, was the Exxon Valdez oil spill that happened off the coast of Alaska. So there's a number of different stories. Some people claim the captain was drunk. I think from what I can gather, they basically hit an iceberg, and then the ship started to capsize and release a great deal of oil into this very pristine environment. And this ended up in the oil from the tanker basically spreading over a huge distance of the Alaskan coast. This is a nice map here showing the scale of this oil spill off the eastern coast. So it's equivalent to essentially Massachusetts to North Carolina. And in this particular incident, 42 million litres of oil was leaked over this huge region of pristine environment. So it was a massive, massive disaster. And this had a number of consequences. Of course, I always like this photo here is of the shoreline covered with oil and the workers who were involved in cleaning up. This particular chap seems to be smoking, which actually would probably be quite safe because the temperatures here would be so low you'd probably have little chance of igniting it. But it was a funny little thing. The damage to the local wildlife was massive. So basically, the otters got all the media attention. You can see this little chap up here is obviously quite distraught by the incident. So about 3,000 otters died, about 300 seals, about 20 whales, and about 250,000 birds. So once the birds got oil in their feathers, they couldn't fly, they couldn't keep warm, and they would die from this. And of course, this affected the local environment, so having oil along your shores is obviously not great for the local residents. This is a local chap getting some oil up from the general environment, and of course, that actually affected local communities both in terms of tourism, but also in terms of fishing. So a lot of these communities along this Alaskan coast relied on fishing for their livelihood. Obviously, they can't fish if there's oil in the fish. So I won't go through what happened with the spill, but I'll just say that after 25 years, there were still some outcomes that were negative from this particular event. So it had a very long-term effect on the local environment and the local communities. So for those of you who are otter lovers, the otters did rebound in population. So you can see these two little chaps up here are quite happy about that. Apparently otters still die if they dig up a clam which has oil inside of it. But one one thing that was a permanent result of this spill, or is believed to be, is that the local herring fisheries in that region collapsed. So they're not sure if it's completely due to the oil spill, but that was probably one of the contributing factors. Now, the company ExxonMobil that actually ran the tanker, they, of course, got a lot of negative publicity, and they lost about $4.4 billion because of this, and that was due to the cost in cleaning up the spill, litigation, and fines. So these type of events can have really negative consequences, of course, for the local environment, but also for the companies that are actually transporting the oil. So that's sort of about 30 years ago, but this is perhaps something that's more fresh in people's mind, and this is the Deepwater Horizon blowout. So this Deepwater Horizon rig here basically was testing an oil field. A huge burst of gas came from 1,500 metres below the ocean, went up here and basically caused the oil rig to explode. So this, if you remember, was that colossal disaster at the time. And there are a number of huge consequences for this. So the first, of course, was that people died on this oil rig. And so 11 very experienced crew died. So this was a group of individuals who'd been working on oil rigs for 7 years. They had no accidents. They knew exactly what they were doing. From what we gather from the reports, basically they made a number of small errors and they were being rushed. And this led, of course, to this. This just had the unfortunate consequences of leading to this particular disastrous event. 17 were injured. In many respects, if you remember the footage of this particular event and just how huge the explosion was, this could have been a lot worse because there were 126 people on the oil rig. Apparently, some of them were actually giving us an award on safety that day, which is slightly ironic, but I guess you can't make this stuff up. So they were lucky enough to escape to a nearby boat. And in many respects, this had similar environmental consequences to the Alaskan spill. So there was huge loss of animal life. Again, I think it was well over 1 million birds from memory. It affected the dolphin population, the seal population, local fisheries, and it was considered the largest environmental disaster in US history at the time. I think it still is. And of course, there were consequences for the companies. So it wasn't just BP or British Petroleum, like Barack Obama liked to call it on his sort of more Anglophobic days. It was also Transocean, also Halliburton that were involved in this. So all these companies obviously got a lot of backlash, and of course they had to pay for the costs. And I'll show you that the costs are quite huge. So this is what happened in the oil spill. The rig was just off the Louisiana coast here, and you had this whole region spread with oil, including this about 2,000 kilometres of coastline. So it wasn't just the ocean that was affected. Again, it was the land, coastal regions, which of course included a lot of wetlands and environmentally important areas. So, what happened and how did they actually fix this situation? So at the time, this particular oil rig was the deepest on the planet, so it went down to 1,500 metres. And when it exploded, oil kept leaking up from here for about 90 days until they could manage to seal the leak. And at the time, the technology wasn't present to do this, so it obviously took a long time to try and develop it and also to bring it down here. So when the oil was released, a large amount actually ended up near the bottom of the ocean. And we're still not sure what the fate of this oil is even 10 years later. But a large amount went to the surface where it floated on top. So what the authorities did at the time is a number of things to try and actually alleviate the environmental consequences of this particular oil spill. So the main thing they tried to do is actually confine it into a certain area, and this was quite difficult given the large amount of oil that was released. And then some of this would evaporate, and then some of it was burnt. Now, a large amount of the oil that was actually couldn't be contained in that area was actually they tried to break it up with dispersants. So these are chemicals which can break up the oil and make it easier for organisms to degrade, which I'll go through in the next slide. And then some of it actually mixed with organic matter and then sank to the bottom of the ocean. And this is called marine oil snow. So this is a mixture of organic matter and oil, which is heavy enough to sink to the bottom of the floor. And this will sort of be what I discuss near the end of the talk. Now this is just a nice, I think, figure just showing the type of wildlife that was affected by this particular event. So people tend to focus on the larger, cuter and friendlier animals, but you can sort of see here that there was sea turtles, birds, wetlands, marine mammals, corals and the fisheries. And in the case of the animals, I think there were about 250,000 turtles that were affected, and everyone loves dolphins, so there was a lot of publicity for the dolphins, but apparently even now the dolphins have reproductive issues in this particular environment. For the local communities, it was the same as in Alaska. Basically, no one wants to swim on shorelines which are covered with oil, so this affected the tourism industry and, of course, affected the fisheries industry. In this case, the fishery industries recovered and they actually developed better environmental practices, so in a strange sort of way, it was a bit of a blessing in disguise. And there was a financial cost. And the financial cost was borne by probably every single person in this room and, in fact, most people in the UK. So for BP, it cost 65 billion as of last year. And prior to this event, BP paid 1 in every 7 pounds in dividends into British pensions. So everyone in the UK who had a pension lost money from this event. And I think it was only the last couple of years have started to sort of get back to those dividend levels. And overall, the cost of this oil spill was 146 billion, according to a paper that came out a couple of years ago. So the point I'm trying to get across is that while oil spills are rare, the environmental financial costs are huge. And so 146 billion is just a number which is unfathomable. So there were some winners. And for those of you who are a fan of John Grisham films, I know that Tommy Lee Jones didn't play a litigation lawyer, he played an attorney general, but I like the film and he looks cool in this. So US litigation lawyers made $680 million. So they were obviously having a good payday for that period of time. It's easy to make fun of lawyers and it's quite fun too. I have a lot of friends who are lawyers and they always have the best lawyer jokes. But scientists did really well out of it too. And so not these particular scientists, I just sort of Googled happy scientists. And quite a lot of images came up actually. I wasn't expecting that. I'm not sure how many would come up if you Googled depressed scientists, but we'll see. But if you spend any time with an academic scientist, they'll start whinging about how hard it is to get grants, and I'll whinge later in this talk. But basically, if you were a scientist who was living in one of the Gulf states, so Texas, Louisiana, Mississippi, Alabama, or Florida, suddenly you had access to a pot of $500 million in grant income over 10 years. So in a way, if there was an oil spill in Britain, I'd be sad about it, but also somewhat happy. But hopefully it won't happen. But that also led to some positive consequences. And the main one was because these companies were so ill-prepared for these oil spills and because the financial cost was so huge, there is now a company in the world called Oil Spill Response, which has a base in Southampton, but also has bases around the world. And what they have is equipment which allows them to respond very quickly to oil spills. So I'm just showing one piece of equipment that allows them to sort of plug a hole like the Deepwater Horizon oil spill. So this means that it's probably less likely that these type of events will happen in the future. And if they do happen, we can basically stop the damage from being as great as it was in the Gulf of Mexico. We also learned a great deal about how important microbes are in actually bioremediating these oil spills. So during this oil spill, I've just got a nice photo here of some oil droplets and some bacteria sitting around them. But during this oil spill, as I mentioned, a lot of it evaporated, a lot of it was burnt off, a lot was confined in an area and pumped away. But according to the National Oceanic and Atmospheric Administration, about half of this was actually degraded by microbes. And so this is a very interesting biochemical process which only a subset of bacteria and some fungi can do. So you need very specialized enzymes and very specialized transport processes in order to be able to degrade hydrocarbons. But of course, hydrocarbons are very energy rich. That's why we use them in our cars. So if you're a bacterium that can degrade hydrocarbons, you have access to a very energy rich source of food. So this will be my only chemistry slide. So apologies if you do not like chemistry. I wasn't sure how many people know what hydrocarbons are. Basically, a hydrocarbon contains, as the name suggests, hydrogen and carbon. And this is pentadecane here, which I'll talk about more in the rest of the talk because it's quite important. So generally, we draw hydrocarbons like these straight lines. And there are a range of hydrocarbons which are found in oil. And so alkanes are like these here. An alkene has a double carbon bond, so it's a bit slightly harder to break apart. An alkane is very similar, it has a triple bond, so it's even harder to break apart. But some hydrocarbons also form these circular rings of carbon, which you can see here, and these can either be single or these sort of multiple rings attached together. Aromatics are similar to these cyclic hydrocarbons, but they have these double carbon bonds. And why the aromatics are important is that these are some of the most toxic compounds that are found in oil, so compounds like benzene. And then you have these sort of large compounds called asphaltenes and resins. So these tend to be a combination of alkane parts, aromatic and cyclic. And basically, when people classify that oil, they classify it into either light or heavy. So light oil tends to have more of these, heavy oil tends to have more of these. And from a biological perspective, there are different groups of bacteria which are capable of degrading certain types of these hydrocarbons. So you'll have one group that degrade these type of hydrocarbons, another group that will degrade these hydrocarbons, another group with those, and these actually tend to be very hard to degrade, so they tend to stay in the environment for a long time. However, they're not very bioreactive, so they're not very toxic to most biological life, unless, of course, it sort of obstructs systems in birds or other organisms. So that's very important, the fact that bacteria can only degrade specialized types of hydrocarbons, and only certain bacteria can do this. And of course, there's different types of hydrocarbons based on the size. So the size of the hydrocarbon will determine its properties in the environment. So carbons less than 4, this is stuff like methane, ethane, butane, these tend to be gases. So if these are leaked from an oil rig, they'll basically just evaporate into the air. You have soluble hydrocarbons, so these tend to be compounds like octane, which is in petrol. So these are water soluble. So when these are spilt in an accident, you actually don't know where they are because they just basically solubilize into the water. They have no sort of visible properties, and basically there's nothing much you can do about them. So what people tend to focus on in oil spills is these larger 8 to 16 carbon hydrocarbons, and these are water insoluble. So if these are released from a ship or from a rig, they'll float to the surface, they'll have the characteristics of oil, they'll look black, they'll be gluggy, and they'll look unattractive. And so these tend to be the hydrocarbons that people are interested in cleaning up in these spills. And then once you get much above 16 carbons, they tend to be solid. And so these can also be an issue. That's why some of the oil looks a bit gluggy and a bit solidified. It has a large amount of these hydrocarbons. So what I'm really going to focus on in this talk, and which is most relevant, are these two hydrocarbons here, the alkanes and the alkenes, and predominantly the ones around about 16 carbons in size. So, as I mentioned, some bacteria have very specialized properties which allow them to essentially degrade or consume these hydrocarbons and use them as a source of energy. So this is just a schematic of how this works. So if we start here on the left, if the bacteria that can degrade hydrocarbons detect the hydrocarbons in the environment, what they'll do is they'll release certain chemicals which act as emulsifiers. So obviously, if there's oil in the environment, it's going to be in the form of large droplets. And from a bacterial perspective, these might be too difficult to deal with. Ideally, they want single hydrocarbons that they can import into the cell. So when they release emulsifiers, they actually break apart the hydrocarbons, and you see sort of an example, there's different types of bacteria here, sort of breaking these oil droplets up into small amounts. So once they have single hydrocarbons, they have specialized transporters that allow them to import the hydrocarbon into the middle of the cell where it can be used. And then once this is done, this is up here, they have specialized enzymes that can convert these hydrocarbons to lipids, and then these lipids can be used as a source of carbon and energy. And in the end, the final product tends to be carbon dioxide. So as I mentioned, and this is very important, only certain microbes can degrade hydrocarbons, and in this case, there's a subset of hydrocarbons that degrade these alkanes of about carbon-16 in size. And what's also interesting about some of these microbes is they are so specialized for degrading hydrocarbons that they are unable to degrade most other carbon sources. So this means they really need hydrocarbons to basically keep surviving. And we call these obligate hydrocarbon degrading bacteria. Now, one would expect to see these obligate hydrocarbon degrading bacteria in heavily polluted sites, so near oil rigs, maybe in ports. But what's really strange is that people for many years have been detecting these obligate hydrocarbon degrading bacteria in sort of minimally or unpolluted environments. Which sort of begs the question of, if they need hydrocarbons to survive, how are they surviving these unpolluted waters? And this is where we get to the algae portion of the talk. So my interests are predominantly in cyanobacteria, which are called blue-green algae. And I won't go into too much detail, but they have some interesting properties that make them different from most other bacterial species. So this is just a schematic here of a cell we drew for a previous paper. So they have sort of the interior of the cell and a cell wall which surrounds this, common to most other bacteria. But they also have a series of internal membranes. So these are internal compartments, and these are for photosynthesis. So cyanobacteria are the only bacteria that can perform oxygenic photosynthesis. And what this means is they have the ability to harvest sunlight, and then they use a large enzyme complex called photosystem II to take that energy and to break apart water. And it was actually this event which was one of the most important in the planet's history, because when they break apart water, they produce energy, but they also produce oxygen. So nearly all the oxygen on the planet is derived from this process. So cyanobacteria were responsible for creating an oxygenic environment that allowed other organisms to basically evolve. And so what they'll do is, during photosynthesis, they'll produce electrical energy, and by various processes, this electrical energy is converted to chemical energy. And the chemical energy is used for many things in the cell, but one of the most important is fixing carbon dioxide, so converting carbon dioxide to sugars. And then these sugars are used for a whole range of metabolic processes in the cell, mostly to help the cell divide and basically biomass. So it's used for biomass production. And the biomass consists of a whole range of chemicals, but what's really strange is that cyanobacteria actually produce hydrocarbons. And these are odd-chained hydrocarbons, that's quite important. They're carbon-15, carbon-17, or carbon-19. So they're alkanes or alkenes containing 15, 17, or 19 carbons. Now, a couple of years ago, we thought cyanobacteria were the only organisms that could do this, but a group in France discovered that eukaryotic algae can do something very similar. So eukaryotic algae, I've just shown a very simple schematic of the cell. They also can perform oxygenating photosynthesis. So in some point in the history of the planet, an organism basically consumed a cyanobacteria, but rather than eating it and breaking it apart, it essentially hijacked its ability to photosynthesize, decided this was a good idea because it's free energy, and then that cyanobacterium developed into a chloroplast. And so a chloroplast works very similar to a cyanobacterium. So they harvest sunlight, they break apart water into oxygen and electrical energy, they make chemical energy, and they fix carbon dioxide into sugars, which then gets fixed into biomass. And so we only thought cyanobacteria could make hydrocarbons because they were the only ones that contained the genes that encoded the enzymes that allow the cell to make hydrocarbons. But a group in France discovered that, in fact, algae have a different set of genes that allow them to make very similar hydrocarbons to the cyanobacteria. In this case, they also make odd-chain hydrocarbons, and so far they've discovered ones alkanes and alkenes of carbon-15 and carbon-17 in size. So this seems to be a very similar process, and at this stage we think they're probably making them for a similar purpose. And just for those of you who maybe don't have a biology background, this conversion of carbon dioxide into biomass using energy from sunlight is called primary production. And I think what very few people are aware of is that they know that trees do this, but, and at a very large scale, but cyanobacteria are responsible for about a quarter of the global primary production, so a quarter of the conversion of CO2 into biomass. And eukaryotic algae are responsible for another quarter, and then plants on the land are responsible for the remaining 50%. So combined, cyanobacteria and eukaryotic algae are as important as land-based plants for carbon fixation in the global environment. So I thought I'd say briefly what I do. So I have a number of different projects, but the majority of my work is dealing with cyanobacteria. So I spend a lot of time investigating the biochemistry and physiology of cyanobacteria, understanding how these important organisms work, basically. And then that feeds into how I understand their role in the environment. So obviously, if these organisms are producing 25% of the global biomass, then they're actually they're probably going to be very common and are going to play an important role in the environment. And the last thing, which sort of dealt with the introductory talk, is I have a great deal of interest in using cyanobacteria for synthetic biology applications. So it's very attractive, the idea that these organisms can convert carbon dioxide into a range of compounds. What we do is we use synthetic biology tools, which I partially develop, and I use some other people's ones, to try and genetically engineer these organisms to produce useful commercial compounds. And so this would basically be a more environmentally friendly way of converting carbon dioxide to industrial compounds and potentially replacing some of the production that currently is derived from fossil fuels. So really, I'm going to just talk about this part today. So I had a strong interest in hydrocarbons in cyanobacteria a long time ago, and there were basically three scientific questions I was asking. So the first one, which I won't talk about today, but is still an ongoing piece of research in my lab, and I've got a large international grant to fund this, is why do cyanobacteria and eukaryotic algae produce hydrocarbons? What physiological role do they play in the cell? And we think they do something that's quite unique in nature. The second thing is, if these cyanobacteria and eukaryotic algae are so abundant in the environment, how much hydrocarbons are they producing? Is it a lot, basically? And if they are producing a lot of hydrocarbons, what are the environmental consequences of that? So, to answer the second question of how much hydrocarbons these organisms are producing, we decided to focus on two groups of cyanobacteria, and these are Prochlorococcus and Caucus. And for those who have never heard these names, they should be really aware that these are actually amongst the most abundant organisms on the planet. So these two groups of organisms alone are responsible for about 1/8 of the global primary production. And the reason we focused on them, apart from the fact that they're very abundant, is other research groups over a very long period of time have actually got fairly accurate estimates of how many of these organisms are in the environment at any one point in time. And this is what these maps here show. So basically what you can see if you focus on the top one here is this is the abundance of Prochlorococcus in marine waters around the world. And you can see here that they're very, very abundant. If you look at the scale down here, the redder it is, the more cells there are per milliliter of seawater. And you can see here that there's actually they're very, very abundant in equatorial waters, and as you go towards higher and lower latitudes, the numbers decrease. And just to give you an indication of how abundant these organisms are, in some waters they can be as high as 1 million cells per milliliter of seawater. So it's quite staggering just how many there are in the environment. And from these studies, they actually determined that at any one point in time, this is averaged over a whole year, there are 2.9 to the 10 to the 27 Prochlorococcus cells in the ocean. Now the other group, Silicococcus, is not quite as abundant as Prochlorococcus. You can see here there's no red on this particular world map, but they're also larger cells, and they tend to be found in more environments. So you can sort of see here that the Prochlorococcus stop at these two latitudes. You start to get more Silicococcus in the polar waters, and they're particularly abundant in coastal areas. And again, people have actually worked out the number of Silicococcus cells in the ocean at any one point in time. And I asked some of my colleagues whether these were accurate, because when you look at the error bars here, we've got 2.9 to 0.1 times 10 to 27 cells. So it seems very accurate estimates, and basically everyone in the field seems to agree with this. So it's quite a staggering piece of work that we've been able to actually work out the numbers of these particular organisms, given how large they are. So what we wanted to know was how much hydrocarbons do these two groups of cyanobacteria produce. So I'm just going to go sort of through the process of how we did this. So the first and actually the hardest part of this is actually growing the cells. So I'm going to show on the right-hand side pictures of people who did the work, which I think is very important. So Steve Biller, what's really important is we need to grow up enough cells to analyse, but they also have to be axenic, which means there can't be any other bacteria in that culture. And so Steve Biller comes from a lab in MIT, and they're one of the few groups that can actually grow Prochlorococcus in pure cultures. So despite the fact that it's one of the most abundant organisms on the planet, it's actually extraordinarily difficult to grow in the lab, and this is a real issue for the research community, but he can actually do it. And then Blanca from the University of Warwick actually sent me some cultures of Cyanobacteria species from the marine environment, and I grew these up. And I'm just going to some examples here. So they're called Cyanobacteria, so they're blue-green bacteria, and this is sort of what their normal colour is, this one on the right here. But what you'll see here is that these are other species, and they have a whole variety of quite amazing colours. So most microbiologists tend to work on species that are dull, brown-looking colours, and they often stink. So I had the good fortune to work on brightly coloured, fancy-looking species, and they don't smell either, which is fantastic. I can tell you that from my PhD, where I worked on a particularly smelly organism. So basically, we grew these up in the lab, and then I got samples sent to me by Steve Biller from MIT. So what we then did next was we basically extracted the hydrocarbons from these cells and then quantified the amount. And for that, we used a GCMS, and Matt Davey, who's at the University of Cambridge, was moving up to Auburn soon, he did this work for me. And so what we did is we knew there were a certain number of cells in our sample. So all we did was quantify the amount of hydrocarbons in that particular sample and then work out how much hydrocarbons there are per cell. So this is the amount. So in the hydrocarbon content per Prochlorococcus cell, keep in mind these are very small cells, was 0.509 femtograms. So if you've not heard of a femtogram, I hadn't heard of it too before this stage, but it's 0.00000000000000509 grams of hydrocarbons per cell. We also did the same for our Cyanobacteria samples. So in this case, we had 3 different Prochlorococcus species. We had 4 Cyanobacteria species, and we did this for each one. This is the average amount from those different species. And we did the same for Cyanobacteria, and we found there was 0.9075 femtograms, and I won't read this out again because, quite frankly, I probably got it wrong, and it was quite boring for everyone involved. So we now know how much hydrocarbon there is per cell, and we know how many of these cells there are in the ocean. So all you do is a simple mathematical calculation. We multiply the number of cells in the ocean by the amount of hydrocarbons per cell. So this seems very simple, but I actually got 5 different people to do these calculations. And there's been a couple of papers which have been retracted in Nature recently because someone didn't carry a 0 correctly. Yes, 2 in the last year. So they were out by tenfold. So in this case, I was absolutely paranoid about making sure the amounts were right. I got the computer to do it, I did it, I got other people to do it. And so the result of these calculations is that we think that in Prochlorococcus cells in the ocean at any one point in time, they've got 1.48 million tonnes of hydrocarbons in those cells. In the case of Cyanobacteria, it's a bit less because there's a bit less cells obviously in the ocean. So we think there's 0.635. And when you add these two totals up, for these two groups of cyanobacteria, there's a bit over 2 million tonnes of hydrocarbons sitting in these cells at any one point in time. Now, of course, these cells aren't static. They're biological living organisms. And so they're dividing. This is just a nice photo of some cyanobacteria, I think some cyanobacterial cells dividing. So they're obviously dividing, they're producing more themselves. But they're also basically being destroyed too. So in this case, I've got a nice schematic of a virus, and there's a lot of viruses that invade cyanobacteria in the environment. And of course, when they invade the cyanobacteria, they produce more of themselves, and then they break apart the cell to release their progeny, and in the process, they'll release hydrocarbons. And then, of course, cyanobacteria form the bottom of the food web, so there's lots of organisms that actually eat the cyanobacteria, but don't have the biochemistry or the enzymes to actually degrade the hydrocarbons. So these will be released from these type of organisms. So there's a turnover rate of these organisms in the environment. And we have some fairly good calculations as to how often these cells divide. And the population seem to stay relatively constant depending on the environment. So we know that Prochlorococcus cells divide every 1 to 2 days. So in this case, what you do is you take the amount of hydrocarbons in your cells at any one point in time, and you divide them by the division rate to determine the amount of hydrocarbons they produce per year. So in the case of Prochlorococcus, we think it's between 269 to 530 million tonnes being produced annually by Prochlorococcus cells. And in the case of Cyanobacteria, these can divide at quite different rates depending on where they are in the environment. So some cells divide every 1 day, some divide every 7 days. So this gives us a much larger range of possible hydrocarbon production by these cells. But if you combine these two totals together, you get this quite extraordinary total of 308 to 771 million tonnes of cyanobacterial hydrocarbons produced annually. And when we got this number, we went back and we actually checked our calculations again because we were a bit paranoid that this was a mistake. Oh, sorry. So, I'm not sure if this number means much to anyone, but I'll actually give you some comparative numbers to give you an idea of the scale of this. And just to keep in mind that this is only for 2 groups of cyanobacteria. So there are other cyanobacteria in the environment which are quite abundant. And this is quite an interesting one here called Trichodesmium, which forms these sort of spiky arrays. And this species is very abundant. In fact, it actually fixes a great deal of the nitrogen in the environment. So this is conversion of nitrogen gas into biologically useful nitrogen that can be used by cells. So they play an enormous role. And of course, we don't have any accurate estimates for eukaryotes, but we know that quite a lot of eukaryotic algae that are in the ocean are highly abundant and likely also produce hydrocarbons. So this total here could be much larger. And we actually did want to go out and test this in the ocean because, of course, this was all done in the lab and it really needs to be verified. So I'm going to have my whinge about grants. Basically, I put a grant to test this in and we didn't get funded, but it'll be a short whinge, don't worry. No one wants to hear anyone whinge. But there was a group in the United States that really liked this idea a lot, and they got funded, mostly because they had better reviewers than I did. I was one of them, who actually had more imagination than the reviewers I got. I'm not sure who they are, but yeah. But anyway, what they found, to cut a long story short, because they haven't published this yet, but I've seen the results, is they actually found when they went out and they quantified the hydrocarbons in the populations of these species in the ocean, that their numbers were very similar to ours. So we're actually quite confident of this result. It's been proven, the numbers from the lab results and the numbers from the oceanographic field studies are very similar. So it gives us a great deal of confidence that these type of numbers we're seeing of hydrocarbon production are real. So to put this into comparison, I've talked about two disasters. In the case of the Exxon Valdez spill, the one in Alaska, the total amount released was 36,000 tonnes, so obviously a hell of a lot less than this. And the Deepwater Horizon spill, which has been the largest release of oil from any single event, human event, was 435,000 tonnes, so a great deal lower. Now, there is a certain amount of oil that's released in the environment just from human error and from ships and such, and this is estimated to be 1.3 million tonnes per annum. And so this is about 100-fold less than what you see from production from these two groups of cyanobacteria alone. So in the brochure, I said that it was similar to Saudi oil production. I've now moved on to United States oil production because the United States has been the biggest oil producer in the world for the last couple of years. So when you look at annual United States oil production from the largest producer on the planet, now we're starting to get to similar numbers, so 712 million tonnes. And then when you look at global oil production in 2019, this is a bit over 4 billion tonnes. So it's quite possible that biological organisms may be producing well over 1 billion tonnes, which is possibly a quarter of the annual global production from all human activity on the planet. So we're actually talking about really vast amounts of hydrocarbons produced biologically. And there are other species that also produce hydrocarbons. So insects produce them, plants do. They're different types, but it does suggest that this type of production is quite common in the environment. So this got a lot of press at the time when I released this. This was only half the story, but clearly journalists only read the first part of scientific papers. So we were quite concerned when this was published, and my collaborators were concerned that the press would pick this up as an excuse to basically pollute the world, because who cares, there's so much hydrocarbon production from biological organisms anyway. But one other thing that we never thought of was that some journalists would think that you could actually harvest oil from these organisms. And so this was a headline in the Daily Telegraph. Thankfully, it's only in the paper version, and it was under the section which had a news story about the Kardashians, so you can sort of see how much importance the paper attached to this study. But basically, they said the catch of the day may soon be petrol, so they were basically implying that we might go out and harvest hydrocarbons. And they clearly didn't, like I said, read the paper beyond the abstract, because while the amounts I've given are quite large, obviously the ocean is quite big. And so the amount produced per mil of seawater of hydrocarbons per day is actually incredibly low, so about 2 to 540 times 10 to the minus 12 grams per day. So we will not be doing this, basically. So if these organisms are producing hundreds of millions of tons of hydrocarbons annually, why isn't the ocean just a giant oil spill? And they're probably doing this for hundreds and hundreds of millions of years, because every single cyanobacteria seems to produce hydrocarbons, and a great many eukaryotic algae species do. And we sort of knew the answer before this. We knew that there are bacteria and other organisms that actually can utilize these hydrocarbons as a source of energy. And we did some experiments showing that some of these organisms can use oil and can use cyanobacterial hydrocarbons. But perhaps a more interesting question is, I mentioned before that we see populations of these obligate hydrocarbon degrading bacteria, so the bacteria that can only really utilize hydrocarbons and very few other carbon sources, and we see them in waters which are not polluted by human activity. So the more important question is, are these cyanobacterial hydrocarbons actually supporting these populations? And this could have impact for oil spills, which I'll show in the next couple of slides. So we did a very simple experiment, and this is the only scientific experiment I'll show in the whole talk, so if you don't like seeing graphs, this is it. And I'll go through it very carefully. So we basically grew up a culture, and we had a proportion of hydrocarbons to cells that we see in ocean environments in these minimally polluted waters. So obviously there's a lot more cells and there's a lot more hydrocarbons in this culture, but the proportions are the same as the lower numbers you see in the ocean and the lower amounts of hydrocarbons you see there too. So I'll go through each of these results because there's quite a lot of information here. So we took a culture and we split it. And we gave hydrocarbons to one part of the culture and we gave no hydrocarbons to the other. So when we give no hydrocarbons to the cells, what we see is a gradual decrease in the cell number. So the cell number here is shown on the y-axis and the time in days is shown in the x. So this seems to suggest that without a source of hydrocarbons, these cells die, basically. Now, when we give them hydrocarbons, what we see is that the number goes up a little bit, and then after 3 days, it comes back down to the initial number. So this shows that these hydrocarbons are supporting this population. It increases a bit over this time period and then comes down again. And if you give these populations no more hydrocarbons, they keep going down. Now, if you give them a new batch of hydrocarbons, once they've used these up, they can then use this one, and you see that this growth continues. So what this seems to suggest to us is that these obligate hydrocarbon degrading bacteria need a constant supply of hydrocarbons, otherwise they'll start dying and the population will go down. And we think it's likely that these are the biological hydrocarbons produced by cyanobacteria and algae. So what we did is we actually we think this is a new biogeochemical cycle that's happening in the oceans, and we called this the short-term hydrocarbon cycle. So in this short-term hydrocarbon cycle, we have cyanobacteria which are using energy from sunlight. They convert carbon dioxide to these alkanes, which are then released from the cell. In turn, these alkanes are used by hydrocarbon degrading bacteria as a source of energy, and they release carbon dioxide, which is then taken up by the cyanobacteria to produce more alkanes. So this is what we call a short-term hydrocarbon cycle, because it likely happens over a period of days. Now, why this might be important in dealing with oil spills is these bacteria, hydrocarbon bacteria, play a role in what we call the long-term hydrocarbon cycle. So when cyanobacteria, algae, and other organic matter sinks to the bottom of the floor, over periods of millions, hundreds of millions of years, this is converted to oil. And so if we release the oil here, if it naturally seeps out of the bottom of the ocean floor, which it does in certain places, or we have an accident and it gets released in the environment from that, these hydrocarbon degrading bacteria can then basically break the oil down, degrade it, and reduce the consequences of that environmental oil spill. So what we think is that these biologically produced hydrocarbons are basically acting as a bit of an insurance policy for the ocean. So it's just a lucky consequence of this particular cycle. So if you don't have these hydrocarbon degrading bacteria in the environment and you spill oil there, then basically there's no biological mechanism to actually degrade the oil. So it will stay in the environment until it evaporates or until it's spread out over a huge area. So by maintaining this population of hydrocarbon degrading bacteria, even though it's quite small, on these biologically produced hydrocarbons, you have a small population that can expand in number if there's a release of oil from, say, a human disaster. And then, of course, these organisms, this is obviously a great day for them because they suddenly have a massive supply of oil, of food. So they'll expand in number, they'll help degrade the oil, and when the oil is all used up, the population will go down to the small size which can be sustained by biological hydrocarbon production. So this was quite neat, and again, we wanted to test this, but the American group actually showed that this seems to be correct, because what they did is they actually went out to these minimally polluted waters, so these sort of pristine waters, and they actually took populations from that environment and they grew it on the range of hydrocarbons. So what they basically were testing was whether these bacterial populations would increase in size depending on the hydrocarbons. In this case, the only group that actually increased in size were the ones fed on the alkanes, which are similar to what cyanobacteria produce. So this seems to suggest that this population is being maintained by that. And in the case of an oil spill, these would be the ones that would degrade at least that proportion of the oil that was released. So just to add that, realising am I over time yet? I'll go through this quickly. I talked a bit about some of the oil that sinks to the bottom of the ocean floor. And obviously the cyanobacteria and algae are in the surface waters because they're photosynthetic, they need light to survive. So we think the hydrocarbon cycling is happening up here. So what about the oil that sinks down here? So what we think is happening is that there are different populations of hydrocarbon degraders down there. And this was a study that was led by a Chinese group, by Zhihao Zhang. And what we were investigating in this particular study was the microbes that live at the bottom of the Mariana Trench. So this is 11,000 meters below sea level. This is the deepest point on the planet, or known point on the planet. So basically what we wanted to know was which type of bacteria live down there and how they're surviving down there, because the pressures are enormous. So it's greater than 1,000 meters pressed against a fingernail. So this is an extreme environment, to say the least. And we're not sure what kind of nutrients sink down there. So this was done with a few people at UEA. So Jonathan Todd, who's a professor in bio, and Nikolai Penduchik, who is a senior lecturer in ENV. So the Mariana Trench is one of those strange environments, and there's not much known about, but there's been a couple of expeditions down there. And funny enough, they found fish at the bottom of the Mariana Trench. So these fish somehow survive at 11,000 meters below sea level, which is just extraordinary. Unfortunately, they've also found rubbish. They found plastic down there. If anyone works for spam, congratulations, your can resist 1,000 kilograms pressed against a fingernail, which is really impressive. Whoever's making this can knows how to do their job, but that shouldn't be there. What we were interested in was the bacteria, of course, and this is just a picture of one of the bacterium from down there. It doesn't look very exciting. But the exciting result was that we actually found the highest proportion of hydrocarbon degrading bacteria on the planet in this environment. And they were greater than 20% of the total number of bacteria there. And we have very few explanations for that. But what it suggests is that if that oil sinks to this part of the ocean, then there are bacteria there that can degrade it. So they could be living on pollution that sort of sinks into the Mariana Trench, because it is a trench and it is where pollution will accumulate. But we also found biologically produced hydrocarbons, alkanes, but these were even. So these had C16, C18 and C20. So these would be produced by a completely different type of biochemistry to what we see in the cyanobacteria and eukaryotic algae. So we think there's other organisms producing these type of hydrocarbons down there, and possibly these are feeding these hydrocarbon degrading organisms we see in this environment. So this is something very exciting which we're pursuing at the moment. And just sort of to end about what we'll be doing in the future is that if we can get the grant money, what we'll be doing is examining the hydrocarbon degrading populations around the UK coast here. And as you see, there's not many Prochlorococcus up here, but there's a lot of silica caucus. And also we want to examine the populations in the Arctic Circle, because this is going to become more of a shipping lane in the future. So what we want to know is if there's an accident in these environments, what are the populations of hydrocarbon degrading bacteria that we can see there, and what is their capacity to degrade this oil if it's released. And this is important because these oil spills still happen. So there was an oil spill last year off the Brazilian coast. It's believed that a tanker was taking oil from Venezuela to Asia. Something happened around there, and about 2,000 miles of Brazilian coastline was contaminated with oil. They still don't know what it is that did it, but the disaster was very much real. So, a bit over time, I think, aren't I? Yeah, sorry about that. So basically, I just want to acknowledge the other people who are involved in the study. So these are all the senior people, Chris, Sasha and Alison at Cambridge. Penny Chisholm was the boss of Steve Biller at MIT. So she's accepting the National Science Medal from Barack Obama. I'm not sure what he's doing these days. I think he's working on his handicap quite a lot. And last year she won the Crawford Prize, which is a Nobel Prize for biologists. And that's for her work in understanding the role of these cyanobacteria in the environment. And David Scanlon, also from the University of Warwick, and these are our sponsors. If you're interested in the papers, these are the two that form the basis of this talk. Thank you very much.
So thanks very much, David. That's been a really interesting story that you've told there, and it has lots of ramifications and lots of, I'm sure there's going to be lots of questions here from the audience. We've got about 10-15 minutes for questions, but there'll be an opportunity afterwards to carry on as well. So over there, if that was the first hand going up, I'll have to move over here because I can't quite see you in the light. One there, please.
I was just wondering whether, given it seems that you're talking about if there is an oil spill, there are bacteria, there are cyanobacteria there that will be able to clean it up. Is there any way that those can be grown in large numbers and kept going in labs so that they could then be dumped in the areas. And
So-
are you doing any research on the ones that can clobber the heavier oil?
So I knew that would be the first question to ask, actually. So there have been people and there have been companies that have worked on this. Now, the issue is that, this is the issue we have with synthetic biology, is that as soon as you take an organism from the environment, you bring it into the lab and then you want to play with it a bit in order to adapt it for a certain purpose, it becomes weaker. And in the environment, of course, it's a warfare out there. You have organisms evolving all the time, and they have to adapt to new viruses, they have to adapt to changing environmental conditions and predators. So to the best of my knowledge, no one's had any real success at introducing foreign organisms into an environment. And I would be, let's just say there was a spill in the Arctic Ocean, for instance. Potentially, if you had organisms you'd taken from that environment and grown in the lab, you could do it. But whether they would survive very long would be the question. And it's something that it may be possible, but the longer you keep in the lab, the less likely it is to work.
Okay, so you wouldn't be able to, for example, keep that, I mean, I'm sure it's a lot more difficult to keep them in some sort of isolated sea pen in various areas along shipping lanes.
Yeah, that's an interesting point. I'm not sure it would be worth it actually at this stage. That oil bioremediation company I mentioned, oil spill response, they're pretty damn good actually. Now they need the natural populations to work, but everyone's been much more careful in the last 10 years since, not surprisingly, since the Deepwater Horizon oil spill. And the case of that Venezuelan one, that was a ship that didn't have any log. They were being naughty basically, or smuggling it out of Venezuela. So in terms of legitimate operations, no one's really made any mistakes for a while. So I think the financial incentive to do this is quite low. And hence the reason I think my grant was failed, because people don't perceive it as a big problem as it once was. Because if there's another oil spill, we'll all be sort of saying otherwise.
One over there first, I think.
I presume the genomes of these bacteria have been sequenced?
Yes.
So are you playing with CRISPR to have a look at what you can do with them?
Oh, well, CRISPR is, I mean, there's a lot of fuss made about CRISPR, but we don't use it for most of our old-fashioned genetic engineering tools work quicker and better. So at the moment I have one surface hydrocarbon degrading species and 3 from the bottom of the Mariana Trench growing on one of my students' benches. And her project is actually to genetically engineer these using old-fashioned plasmids and homologous recombination, if you know much about this. So CRISPR it's not really been as popular with bacteriologists as it has been with other groups of organisms. So we can do very precise genetic engineering of cyanobacteria using the old tools, and nothing has come along in the CRISPR field which is better yet.
David, thank you. It's a fascinating lecture. There's a lot of methane at the bottom of the ocean. Is that in some sort of steady state being created and consumed by bacteria? Because Trump was talking recently about actually exploiting that, and the Chinese already are, which sounds pretty scary. But I wondered whether that is in a steady state, or is it actually trapped in there somehow? Well,
there's of course organisms that produce methane and those that degrade them. And the ones that degrade them are called methanotrophs, and they're very common in the environment. I don't know how common they are down the bottom of the ocean, actually. So I can't answer that question for you, is the answer, I'm afraid to say. I expect that they are, if you've got methane seepage in an area for a long period of time, there's going to be organisms that degrade it there. They're not going to not take advantage of that energy source. And the methanotrophs, a lot of them have adapted just to methane as well. So that's sort of bottom layer of oil from the Deepwater Horizon oil spill. They have isolated hydrocarbon degrading organisms in that particular part of the spill, and that contained a lot of methane. So there were definitely methanotrophs there. Whether they're common in sort of minimally polluted waters is a different question.
One over on this side over here.
Someone at the back.
Oh, one at the back, sorry.
Yeah, hi.
I can't see you up there.
There was a lot of excitement a few years ago about plastic munching bacteria
yeah.
found on a Japanese dump originally.
Yes.
Are they related, these bacteria, to the ones that you're studying?
Not related so much, but a lot of the, some of the biochemical processes similar, but an organism that can degrade hydrocarbons definitely has different biochemistry to one that degrades plastic. And the plastic degraders are quite slow at degrading it. Whereas these guys, you can have, if you start off with a small culture, they'll be growing like crazy in a couple of days. You can make kilograms of them. So it's different biochemistry, and the time scale of that process differs hugely. So it will take a lot longer for these organisms to degrade plastic. Now, if there's a lot of plastic in the ocean, maybe they'll adapt to this. But I think it's quite difficult for them to break apart the polymers in plastic.
There's another one up there, isn't it? Yes?
Oh, thank you. Yes, you've highlighted some very useful organisms on the planet, and the carbon fixing side of it sounds very interesting too, given that we're trying to reduce carbon in the atmosphere. Would there be more grants available to look at that side of it? And given that the ocean temperatures might rise, might they encourage more carbon fixing in the oceans?
So to answer your first question, so there was a lot of interest 10 years ago, so I was actually funded by Shell at one stage to engineer cyanobacteria to produce biofuels. Now the main issue is you have to make money. And so focusing on biofuels was always stupid, quite frankly. No offence if anyone's from Shell, I apologise. But we are focusing on higher value chemicals. So potentially, yes, there is, well, with cyanobacteria and algae, you can grow them on non-arable land with minimal nutrients and seawater. So there is the potential for renewable production from CO2. It's just commercially, you've got to get it to work. So one of the chemicals we're focusing on is $10,000 a gram, whereas biofuels are like 50 cents a litre. So obviously that was never going to work very well. Sorry, I forgot your second question, actually. Apologise.
Well, given that ocean temperatures are rising, is that going to encourage?
All right, so this is, thankfully this is not my area, because the people in this area are fighting like mad. So it's the same debate about whether carbon dioxide levels will help plants grow better. And some people actually say it's going to be worse for them. In the marine environment, the temperature's changing. There's lots of papers, and they tend to get published in very high journals. And so there was a paper that came out that suggested that cyanobacteria will do better under these warmer conditions. And then everyone just savaged it. So I would not even begin to make a prediction about this. It could be that the relative proportion of cyanobacteria to, say, eukaryotic algae will change. But then these organisms adapt very quickly. And they often occupy different sort of zones in the ocean. And the diatoms and the eukaryotic algae, they're the most common, actually tend to explode in number when there's a bloom. So when there's a sudden inburst of nutrients into that environment. Whereas the cyanobacteria, especially Prochlorococcus, they tend to be there all the time, and they can survive on very low nutrient sources. So it's a good question, but I think it's a nature question, basically. And no one's answered it yet.
Have you looked at more regular hotspots of oil kind of release, such as ports and harbours, and whether that's supporting populations of cyanobacteria?
Supporting populations of hydrocarbon degraders. Oh, yeah. Yeah. No, that was done before this study. So there were lots of studies. They actually isolated a lot of these organisms from heavily polluted port regions. So in that case, if you've got a lot of human activity and a lot of oil being spilt in the environment, there's always populations there living off that particular source. And the same in the Gulf of Mexico. This is also a very heavily polluted environment from the amount of shipping, and because there's oil rigs everywhere. So in this case, if you have an oil spill there, there's always going to be populations that are going to quickly be able to sort of consume the oil. And in some respects, it's actually a good place to have an oil spill. Now, if you have it in a pristine environment where there's no human activity, this is where we think the cyanobacterial alkanes become important. But then if you have an oil spill there, chances are, if you've got the microbes living on that, they're only going to degrade the alkanes in the oil. And these other hydrocarbons may actually stay there for a very long time, and that's actually what we think happens.
Another one here?
Please forgive me, this is quite a basic question, but what do the cyanobacteria use their hydrocarbons for? Why are they making them in the first
Actually, that's not a basic question. That's actually the really good question. So I got a $1.2 million US grant to answer that question. So it's a good one. So I published one paper on this, and I'm working with people in Imperial College London, a group in Sweden, and a group in New Zealand to answer it. So what we think happens is, for a cyanobacterium, making a hydrocarbon is actually a really good... They have lots of carbon, they have lots of energy, and it takes a lot of energy to make a hydrocarbon. But they can do this because they have both those things in abundance. What they tend to be lacking is phosphorus and nitrogen. And what we think they're doing, and we have fairly good evidence for this, is they actually accumulate the hydrocarbons in their membranes. So they have two sets of membranes. They have an outer plasma membrane, which surrounds the cells, and then they have these internal thylakoid membranes inside the cell, which is where photosynthesis occurs. And the natural state of a membrane is to be flat. But in order to fit these thylakoid membranes in the cell, they have to curve them. And this is quite an important process in nature, and most organisms do it by something called asymmetrical lipid packing. So they have a certain type of lipid in one of the... So the membrane's made up of two layers of lipids. In one of these layers, they'll have more of a certain type of lipid, and the chemistry of that lipid makes the membrane curve. Or they have proteins which help curve the membrane. What we think is happening, and we're always like 99.9% certain of this, is that the hydrocarbons we've shown sit in the middle of the membrane, and when they accumulate, they also induce the curvature of the membrane. So if we actually make a mutant devoid of hydrocarbons, what happens is the cells are larger, they grow poorer, and the membranes are less curved. So we think the hydrocarbons are used by the cell to curve the membranes, to pack them all into like a small area. In the case of those Prochlorococcus cells, they're amongst the smallest bacteria on the planet, so they're 0.6 microns in diameter, but they have these thylakoid membranes. They also have the largest amount of hydrocarbons. So this is our theory at the moment. And if this is true, this is a completely novel way of doing this process in nature.
Well, that's great, David. Thank you all. Thanks for a really exciting seminar. I'm sure there's lots more questions, but there's the opportunity to socialise with David outside and pepper him with all those extra questions. But will you join me now in thank you, David? We're really interested.